Hi, my name's Josie South. I'll be discussing the dynamics of the redclaw crayfish invasion in the Zambezi Basin. I have a background in global change ecology, having worked on invasive species and climate change in marine systems. I've been a postdoc at the South African Institute for Aquatic Biodiversity for 2 years now. Here I work on assessing and predicting the impact of freshwater invasive species in Southern Africa. A quick intro to some of the team working on the crayfish project. Taku, who is completing his PhD on crayfish at SAIAB. Craig, a master's student working on fisheries in the Zambezi at SAIAB. Olaf, who is the chief scientist at SAIAB. And Bruce, our WWF counterpart and Kabompo landscape manager. Now you've met the team, it's time to meet the study species. The redclaw crayfish, Cherax quadricarinatus, is native to Australia and Papua New Guinea. It was initially introduced into Africa for aquaculture purposes. It was then spread through southern Africa via further translocations, as well as aquaculture escapes. The redclaw crayfish have been present in the Upper Zambezi Basin since 2001. However, in 2014, a new invasion was reported in the Brotsi floodplain in Zambia. There have been no strategic assessments of this species until now. Our objective was to determine the extent of the invasion, get abundance estimates, and use the information to start assessing for possible impact upon native freshwater crabs. These represent the closest trophic analogue in African systems. First, to get a bit of scale and introduce our study area, the Upper Zambezi runs from the headwaters in Munilunga, around here, into Victoria Falls, where it then flows into Lake Kariba in the Middle Zambezi system. This is my crayfish travel guide to Southern Africa. We're based in the Eastern Cape of South Africa, and these surveys make up around 4 months worth of fieldwork, which involves driving our trusty alley boat through the Kalahari Desert, all the way into the Okavango Delta. Across this survey, we used a standardized trapping gear and deployed 1,061 traps in total, across 10 systems in 4 countries. Crayfish were detected in the Brotsi floodplains, the Kafue River and Kafue Flats, and in Lake Kariba. Across all of these systems, detection probability was similar. Luckily, there were no crayfish detected in the Okavango floodplain ecoregion in Namibia. However, some individuals have been reported as far down as Sinanga in Zambia. This is a cause for concern as the Okavango floodplain ecoregion is connected seasonally to the invaded Zambezi system through the cylinder spillway during times of high flood. Therefore, it's a likely contender to be invaded in the coming years, which will pose a threat to the currently pristine Okavango Delta. Similarly, upstream there were no crayfish detected in the Kabompo and West Lunga headwater systems. The rate of spread was calculated for the Brotsi floodplain, and this was found to be around 49 kilometers a year downstream and 12 kilometers a year upstream. Catch per unit effort, or CPUE, was used as a proxy for abundance. Here, the large circles represent site aggregated CPUE, while the smaller points show the number of crayfish individuals per trap. The CPUE in the Brotsi floodplain was significantly lower than in the Kafue and in Lake Kariba. However, there was no difference between CPUE in the Kafue and Lake Kariba. There was a clear invasion core to front trend observed, with sites of high abundance panning to sites of declining abundance up and downstream. This is presumably a result of a combination of natural dispersal and habitat suitability, as well as different dynamics on the floodplain ecosystems. There was also a clear pattern in population morphology, where there were significant differences in both carapace length, i.e. CL, and mass between all systems. The Kafue had the largest and heaviest crayfish, while the Brotsi floodplain had the smallest and lightest crayfish. This is likely related to invasion time, as the Kafue population is the most established, whereas the Brotsi population has only been present for 8 years. Regardless, in any of the systems, the invasion has been allowed to proliferate unchecked, for the entire duration. Extremely little is known about African freshwater crab ecology, and yet they represent important and threatened shredder species. As mainland Africa has no native crayfish species, the bottom-knotted crabs represent the nearest trophic analogue. As such, this means that they're a good contender for either being outcompeted by the crayfish or for showing biotic resistance towards the crayfish. Freshwater crabs were a regular bycatch in the crayfish traps, so we used this opportunity to develop a database on crab abundances and distributions between invaded and currently uninvaded systems to fill a data deficiency as well as fill a baseline assessment for future monitoring. A binomial generalized linear model was used to see whether the probability of catching a crab in a trap was related to presence or absence of crayfish and whether there was a relationship with crayfish abundance. There was no relationship detected between crab presence or absence and crayfish. The data suggests that crab abundance is actually very naturally low in these systems and potentially it's strongly correlated to habitat type. We took the same morphometric measurements for the crabs collected and calculated length-weight relationships for both crayfish and crabs for each system. This dataset also includes the invaded Komati River system in South Africa, as well as the uninvaded Buffalo River system in South Africa. The length-weight relationships were used to calculate body condition indexes for each measured individual. Here, scores lower than 100 indicate a below expected body condition, and higher than 100 indicate a better than expected body condition. Again, there were no differences in crab body condition depending on invasion status, where here the invaded systems are shaded in red and uninvaded systems are shaded in blue. Crayfish all had expected body conditions, except for females in the Barotse floodplain, which were in better condition than expected. This may be due to invasion core dynamics and increased reproduction investments in a young invasion. However, this warrants a lot of further monitoring to understand how this will affect population dynamics in the future. So, field data currently suggests that crabs are not impacted in terms of presence, absence, or body condition by the crayfish invasion. Evidence for biotic resistance is therefore currently quite positive. A trait-based proxy of performance was measured in the lab. We used claw closing force as a comparative trait for performance between the freshwater crab Potamnotes pilatus, the redclaw crayfish, and another invasive crayfish, Procambrus clarkii, the red swamp crayfish. Claw closing force can be related to the outcome of agonistic contests, as well as capacity for handling food items. The claw closing force was significantly related to mass in all species, and female crabs had significantly higher closing force capacity than both crayfish species. However, male crabs had similar closing force to male redclaw crayfish. This data indicates that the heavier the animal, the more likely it is to win an agonistic contest. However, this varies depending on both species and sex, and this should also be ground truthed by actual contest experiments, as there's a likelihood for dishonest signaling. Now, to discuss some human dimensions of the invasion, the invaded systems all represent important and heavily exploited fishing areas, with static gill nets being regularly used. However, crayfish depredate the catches and get entangled in the nets. This can cause economic losses both in terms of gear destruction as well as catch destruction. This often results in the fishers throwing the whole nets back into the water or discarding them elsewhere. We completed a scavenging experiment to quantify how much dead fish could be consumed in a 15-hour period per gram of animal. The 15-hour period represents the usual gear soak time. Again, we compared the redclaw crayfish, the red swamp crayfish, and the freshwater crab Potamnotes pilatus at 2 different temperatures. The redclaw crayfish were able to consume significantly more at the lower temperature than the crab at either temperature. So this suggests a high relative impact of redclaw crayfish scavenging regardless of temperature. Similarly, the red swamp crayfish was able to consume significantly more than the crab. This pattern becomes an issue when considering the patterns of abundance seen in the field, as when the crayfish are present, they are more abundant than crabs, which then increases their impact on fisheries further. The panels on the side show the areas in which the crayfish generally consumed. The red swamp crayfish usually consume parts of the abdomen, whereas the redclaw crayfish generally consume parts of the abdomen and tail. However, the crabs mostly went for the abdomen as well as the fish's head. This scavenging behavior might be inciting social change in fishing practices to reduce economic losses. Because of the different areas in which the species consumed of the caught fish, this changes whether the fishers can actually sell their catch. However, the changes in response to this might include starting to dragnet more and also proliferates the use of mosquito net fishing. This could confer secondary ecological impact to the systems. If the native fish species have poor recruitment as a result of crayfish impact and secondary effects of fishing, it's possible that the local communities start to rely on consuming or selling the crayfish. This could then cause the crayfish to become a conflict species in terms of conservation and stakeholder priorities, which would further complicate management strategies. The second phase of monitoring will focus on developing cohesive transboundary policies on crayfish biosecurity by providing a strong evidence base on the economic and ecological impacts. This will include cost estimations of both management and impacts, as well as trying to implement a long-term monitoring program which incorporates native species conservation ecology. This project is funded by DOB Ecology and WWF with support from Namibian and Zambian fisheries departments, as well as NRF SAIAB. I am currently funded by the Centre for Invasion Biology, and Taku and myself are also funded by the DSI NRF Saatchi Chair in Inland Fisheries and Freshwater Ecology. My name is Josie South. You can find my contact information here. Please feel free to get in touch. Thank you for listening.